Hello everyone, I'm Dr. Prashant, and in this presentation we will be talking about innate and acquired immunity. Innate and acquired immunity is part of the medical curriculum of various institutions around the world, but as soon as we hear this topic we are always like, however, through a few concepts and animations we'll try to make this topic simple and easy to understand. The immune system is a scheme of antigen recognition through specialized receptors. Immune memory is required to mount a rapid recall response when the same antigen is encountered at a later time. Finally, the third feature of the immune system is immunologic tolerance. We'll talk about the innate immunity first. Innate immunity consists of pattern recognition receptors, antimicrobial peptides, cells, complement system, and cytokines. The pattern recognition receptors could be transmembrane proteins, in which case they can be toll-like receptors or C-type lectin receptors. The C-type lectin receptors are further subclassified into collectins, pentractins, and MSCs. All these collectively deal with opsonization. The process of opsonization is the process of making antigens more tasty to the immune system by coating the antigen with proteins called opsonins. Pattern recognition receptors may also be cytoplasmic receptors, in which case they could be retinoic acid inducible gene receptors or NOD-like receptors. This is a pathogen, and these are the pathogen-associated molecular patterns or PAMPs. This is an immune cell with pattern recognition receptors. We must keep in mind that pattern recognition receptors are present on dendritic cell, macrophages, monocyte, neutrophils, and epithelial cells. Once the pattern recognition receptor identifies the pathogen-associated molecular patterns seen here in blue, the pattern recognition receptors attach to these PAMPs. This results in chemokines, cytokines, and immune cell recruitment, and finally the process of inflammation, which functions to fight the pathogenic invasion. Toll-like receptors are present on macrophages, dendritic cells, and B lymphocytes. They may be akin to a toll-like structure as seen here. Toll-like receptors are present, as already mentioned, on these components of the immune cells, and they attach on their surface the pathogen, which can bring about bacterial killing through specific antigen-specific cells. Effector cells of the innate immunity are dendritic cells, monocyte macrophages, large granular lymphocytes, natural killer cells or NK cells, and neutrophils, eosinophils, and basophils. Neutrophils, eosinophils, and basophils are amplifiers and effectors of the innate immunity. The neutrophils contain azurophilic granules, which through enzymes myeloperoxidase, lysozyme, and elastase, and specific granules which contain lactoferrin and collagenase, fight infection and contribute to bacterial killing and phagocytosis. Let's take an example of the throat mucosa with the blood vessel as shown. This is a pathogen which is about to invade the throat mucosa. At this point, vasodilatation occurs. This is followed by recruitment and release of neutrophils. When the neutrophils encounters this pathogen, it is taken up by the neutrophil by a process called phagocytosis. Inside the neutrophil, the pathogen is associated with another structure, the phagosome, which on combination with the pathogen forms the phagolysosome as shown here. Inside the phagolysosome, there occurs release of hydrolases, nitric oxide, and reactive oxygen species. This results in killing of the bacteria. However, the antigens from the bacteria are released and carried away into the lymphatics. Dendritic cells could be myeloid, and they could be of the monocyte macrophage variety or tissue-specific dendritic cells. And both of these upregulate the MHC class II expression. We will talk about MHC class II later in this presentation. Dendritic cells may also be plasmacytoid, in which case they activate natural killer cells and pathogenic killing through this pathway, and also activate the mafia of the immune system, the CD8 cytotoxic T cells. The monocyte macrophages are precursor cells from the bone marrow and may also be specialized tissue macrophages. They are the first line of defense and destroy microorganisms through hydrogen peroxide and nitric oxide. They are also capable of cross-presentation of the antigen and secrete interleukin-1, TNF-alpha, interleukin-6, interleukin-12, and chemokines. Monocyte macrophages are also involved in antigen non-specific lytic activity. This is a macrophage or an antigen-presenting cell. This is a pathogen with its constitutive antigens, as we have seen earlier in this presentation. The macrophage now engulfs the pathogen, but the macrophage's handling of these pathogen is different. The macrophage is more professional in handling these pathogens. After digesting a cell, this blue triangle that you see here is the antigen which is left behind from the pathogen. The antigen is now cleaved by proteases and sent to the endoplasmic reticulum for processing. For a moment, we'll move away from the antigen and focus on the nucleus of the macrophage. Inside the macrophage, on chromosome 6, there are genes which encode for diverse proteins. These form mRNA, which eventually transcribe a protein, which is shown with this red graphic. This red image or graphic now combines with our antigen and is now pushed to the surface of the antigen presenting cell. This red graphic is the major histocompatibility complex for which Baruch Benosarov won the Nobel Prize in the 1970s. We'll now shift to the T cell. This is the T cell receptor with its alpha and beta subunits. We have already seen that the antigen is now on the surface of the macrophage in context of the major histocompatibility complex. That complex, that is the antigen, the MHC and the macrophage, will now bind to the T cell receptor. This binding is made more stable by a molecular glue, which is cluster differentiation protein number 4 or CD4. At this time, the T cell receptor releases interleukin-1. Also, there has to be a co-stimulation between these two cells to prevent autoimmune conditions wherein self-antigens are recognized and antibodies are produced against them. This co-stimulation occurs through this molecule, the CD28, and from the macrophage side, this molecule, B7-1. This has to be present if we have to prevent antigen processing, recognition, and antibody production against self-antigens. We'll now move inside the surface of the T cell. Once the macrophage along with MHC and antigen bind to the T cell receptor, genes for interleukin-2 and interleukin-2 receptor are activated. This results in the interleukin 2 molecule binding to the interleukin 2 receptor. This results in clonal expansion of T cells and subsequently expansion of B cells. This is the basic mechanism of expansion of T cells and B cells in recognition to an antigen. Large granular lymphocytes are generally major histocompatibility complex unrestricted in that they don't require the MHC to present an antigen. They can act through their granules to kill pathogenic invasion. The Fc component binds to the opsonized cells. This is present on the surface of the large granular lymphocytes, and they can also kill transplanted foreign or virus infected cells. Also present in the body is the complement system. The complement system can be classical, mannose binding lectin, or alternative activation. The classical pathway occurs due to antigen-antibody immune complex. The MBL pathway occurs because of microbes with terminal mannose groups. And the alternative activation pathway, which is constitutionally always active in our body, occurs because of bacteria, fungi, viruses, or tumor cells. Classical pathway proceeds through C1q, C1r, and C1s. The MBL pathway proceeds through MASP and MASP2, which is mannose-associated serine protease and mannose-associated serine protease 2. On the other hand, the alternative activation pathway proceeds through an opsonin called C3. It is the C3 which is left on the surface of antigens to make it more susceptible to phagocytosis. All three pathways eventually converge on this common molecule, which is C3. The terminal pathway is as follows. C3 is an anaphylatoxin and an opsonin. It causes lymphocyte activation and also itself converts into C3b, C5, and poly C9, which is the membrane attack complex. This results in cell lysis and membrane perturbations. The adaptive immune system constitutes immunological priming, which also results in immunologic memory. T cells arise from the bone marrow, and from there they are sent to the thymus for maturation. From the thymus, they home to the lymph node, where they stay and wait and act in case of a pathogenic attack. B cells, or bursal equivalent or bone marrow derived cells, constitute 10 to 15% of the human peripheral blood. 20 to 30% of lymph nodes constitute B cells, and 50% of splenic lymphocytes constitute B cells. 10% of bone marrow lymphocytes are bursal equivalent or bone marrow derived cells. B cells can recognize unprocessed antigens and have receptors for the complement system. Their main function is to produce antibodies. This is an image showing the immunoglobulins with its heavy chain, light chain, and antibody binding region. This area where the antigen binds is called the idiotype. This is a B cell along with a B cell receptor. This is an antigen which has bound to it. The antigen is internalized and presented on the surface of the B cell in context of a major histocompatibility complex. This is a T cell with its receptor, as we have seen earlier. The B cell now presents to the T cell the antigen. This time, the co-stimulation is taking place through CD40 and CD40 ligand, and it is now the T cell's turn to stimulate the B cell with interleukin 2, 4, and 5 to produce antibodies. We will now move on to the clinical evaluation of a patient's immune function. In general, defects of cellular immunity will cause viral, mycobacterial, and fungal infections, Antibody deficiencies will result in strep pneumoniae and Haemophilus influenzae. Disorders of phagocyte function will cause recurrent skin infections, and deficiency of early and late complement system will result in autoimmune conditions and recurrent Neisseria infection. Immunotherapy may consist of cytokines and cytokine inhibitors, monoclonal antibodies to T and B cell, tolerance induction, and intravenous immunoglobulin or IVIG. Cytokine and cytokine inhibitors are TNF alpha, which consist of antibodies against TNF alpha, namely infliximab, adalimumab, and golimumab, used widely in rheumatological conditions like rheumatoid arthritis and inflammatory bowel disease. A soluble IL-1 receptor antagonist is anakinra, used in certain hereditary periodic fevers and also in gouty arthritis. Antibodies against IL-6 and IL-11 are used in rheumatoid arthritis and other rheumatological conditions. The antibody name is tocilizumab, and interferon gamma is used in the treatment of chronic granulomatous disease. Monoclonal antibodies to T and B cell could be rituximab, which is the anti-CD20 antibody used in hematologic neoplasms, graft versus host disease, autoimmune diseases, and kidney transplant. OKT3 monoclonal antibody against human T cell is used in renal transplant patients and has fewer side effects than horse anti-thymocyte globulin. Interleukin 2 and CD25 targeting antibody is basiliximab for graft versus host disease, and leukocyte migration is prevented by the integrin inhibitor natalizumab, used for multiple sclerosis. Tolerance induction is through CTLA4. We had seen earlier that B71 must bind here to the CD28. However, if CTLA4 binds to CD28, then B71 cannot bind at this location. Intravenous immunoglobulin is used in Kawasaki's disease, graft versus host disease, Guillain-Barré syndrome, and chronic inflammatory demyelinating polyneuropathy. The IVIG targets T cells, cytokines, immune cell trafficking, and complement and Fc receptors. The mechanism of action of IVIG is as follows. We had seen earlier that the antigen is presented by the macrophage to the T cell receptor in this fashion. However, when IVIG is present, this antigen presentation may not occur. Furthermore, we had seen that the B cell produces antibodies like so. In presence of IVIG, B cell will not produce as many antibodies. Finally, in Guillain-Barré syndrome, the antibodies cross the blood vessel through the blood-nerve barrier into the nerve, where they cause demyelination and conduction block. With IVIG, however, they may not be able to cross the blood-nerve barrier. The mechanism of action of IVIG is also complement inactivation, Fc-mediated receptor activity inhibition, and antagonism to pro-inflammatory cytokines. That's it for our presentation on acquired and innate immunity. Thanks for watching, and I will see you in the next video.